Welcome to Inside Drug Discovery. This video describes the discovery program for AZD9574, a CNS-penetrating selective PARP1 inhibitor. The discovery program is summarized based on a Drug Annotation article in the Journal of Medicinal Chemistry. The article was published by scientists at AstraZeneca. A link to the article can be found in the video description. As is often the case, we'll start this discussion with the intended target, PARP1 or poly-ADP-ribosyltransferase 1. PARP1 is one member in a family of PARP enzymes that catalyze the ADP ribosylation of proteins and affects a range of processes. PARP1 inhibition is a known mechanism of action for treating BRCA or BRCA-mutant tumors. BRCA stands for breast cancer. BRCA is the breast cancer gene, a tumor suppressor gene. BRCA mutations increase risks for breast, ovarian, pancreas, and prostate cancers. Many PARP1 inhibitors mimic NAD+ binding, as NAD+ is a cofactor for PARP1. Let's see some of these reported PARP1 inhibitors. Here are 4 PARP1 inhibitors. For each we have PARP1 IC50, PARP1 PARP2 selectivity, a BRCA2 mutant cell assay IC50, an efflux ratio in MDCK cells with high MDR1 and BCRP expression, and the brain-to-plasma unbound plasma coefficient. These compounds show varying selectivity for PARP1 over PARP2, as well as potency in the BRCA2 mutant cell assay. As a group, these compounds are substrates of MDR1, multi-drug resistance protein 1, and BCRP, breast cancer resistance protein. These proteins are transporters that play a role in efflux, which can reduce CNS penetration of a compound as measured by the brain-to-plasma equilibrium constant. Low CNS penetration is a concern with breast cancer, as brain metastasis is often observed in breast cancer. At this point, we have framed the AstraZeneca discovery program and can understand the program's goals. The program started with AZD9305, compound 7, as a lead, a compound with high PARP1 potency, selectivity, and cellular activity. AZD5305's weakness is its high efflux, which reduces CNS penetration. Reducing efflux without overly upsetting the favorable properties is the main objective for the discovery program. Optimization efforts emphasize the amide groups in AZD5305, and we'll start with the amide on the left. On this slide, we have 4 compounds. One is AZD5305, compound 7. The other 3 are new analogs with changes around the left side amide group. You can see some of the changes that the researchers explored, changes to the ethyl group in compound 7, see the new ring, in compound 9, and changes to the neighboring ring nitrogen, removing it in compound 10 and moving the nitrogen in compound 11. All compounds had lower efflux, which is promising. Compound 10 and 11 were both attractive. Compound 10 gave the best CNS penetration, an important objective for the program. Compound 11 improved CNS penetration, albeit to a lesser degree, while also having better PARP1 selectivity. I do not have a column for intrinsic clearance, but compound 11 had a lower intrinsic clearance in human liver microsomes, around half the clearance of compound 10. In the end, the changes in compound 11 were advanced for optimization around the amide on the right side of the structure. Attempts to completely remove the amide on the right had mixed success. Some properties were improved, but at too great of a cost of potency in the BRCA2 mutant cell assay. The researchers therefore focused on changing substituents around the attached pyridine ring. Some of those efforts are on this slide. All compounds shown here are analogs in the 6th position of the pyridine ring. Compound 18, the 6-fluoro analog, had excellent cellular potency, exquisite is the word used by the authors, and an efflux ratio of just 2. Unfortunately, the lower efflux ratio did not translate into dramatically different CNS penetration. In another series of compounds, which are not shown here, the MedChem team had observed higher CNS K values of 0.2 or greater. The researchers kept the 6-fluoro group in compound 18 and turned their attention again to the left amide system to pursue further gains in CNS penetration. Here are 3 final compounds. 18 is from the previous slide. Compounds 22 and 23 show some minor tunings around the amide. Compound 22 had better cellular potency and CNS penetration. To a lesser degree, compound 23 also showed improved CNS penetration. A weakness for compound 22 that is not captured on this table is solubility. Compound 22 was less soluble than compound 23. Ultimately, the collective properties of compound 23 led to its being favored over 22. These are the experimental PK properties of compound 23 across several preclinical species. I did not list intrinsic clearance on the previous slides for space reasons. The researchers, however, did monitor metabolic stability and intrinsic clearance throughout lead optimization. Compound 23 had a quite low intrinsic clearance in human liver microsomes, and the PK data align with this observation. Compound 23 has a fairly long half-life across all species and excellent oral bioavailability. Based on these data, achieving high in vivo exposure of compound 23 for both efficacy and toxicology studies was not a concern. Compound 23, with a designation of AZD9574, was tested for in vivo efficacy in a mouse xenograft model with a BRCA2 mutation-bearing tumor cell line. The results here show tumor growth inhibition observed at the end of the 5-week study. At a dose of 3 mg per kg, AZD9574 reduced tumor growth by 46% relative to vehicle. The 10 and 30 mg per kg groups had similar reductions of around 70%, perhaps indicating a plateau of activity. AZD9574 was well tolerated with minimal reduction of body weight. AZD9574 is currently in clinical trials. On the screen is a screenshot of the only trial listed on clinicaltrials.gov for AZD9574. Enrollment for this study includes patients with breast, ovarian, prostate, or pancreatic cancers. As was mentioned at the start of this video, these types of cancers with BRCA mutations are often treated with PARP1 inhibitors like AZD9574. That is the story of the discovery program of AZD9574. AZD9574 is a highly selective PARP1 inhibitor with high CNS penetration. AZD9574 holds promise for the treatment of multiple oncology indications. Thank you for watching this video. If you have any comments or questions, please leave them below the video. Also consider liking the video or subscribing to the channel. Take care.